So good morning. So last class we have discussed about how protein synthesis occurs or synthesis of protein how it is synthesized. So today we will discuss about that proteins whatever it is synthesized, how that it will enters to the or it will deliver to the target organ. It will enters or it will transport to the target organ. So that is called as protein targeting. That means the process by which proteins are selected and delivered to their destination. That is called as protein targeting or protein sorting and protein targeting. So very important pathway, two pathways is there. Listen carefully. Hello, listen carefully. So very important pathway, two pathway, one is cytosolic pathway, another pathway that is rough endoplasmic reticulum pathway. Okay. So how? Through rough endoplasmic reticulum, that protein will be enters and it will deliver to the target organ, and another pathway that is cytosolic pathway, from cytosol to how it will deliver to different cellular organelles. So in cytosolic pathway, the destination is there, destination or target organ, which is the target? Target is mitochondria, nucleus, peroxisome and cytoplasm. Rough endoplasmic reticulum pathway, it will be target will be your endoplasmic reticulum, membrane of the endoplasmic reticulum, Golgi apparatus membrane, plasma membrane and lysosome. Okay, understood? So two pathway, through two pathway, that protein will be target, protein will be destination. One is your cytosolic pathway, another one is the rough endoplasmic reticulum pathway. Through cytosolic pathway, one is mitochondria, nucleus, peroxisome and cytoplasm. Their target is through cytosolic pathway, target is this, and through rough endoplasmic reticulum pathway, target organ is or target cellular organ is, that is endoplasmic reticulum membrane, Golgi apparatus, plasma membrane and lysosome. Understood? So how that will be target or a destination occurs, slowly I will discuss about. First we will discuss about rough endoplasmic reticulum pathway. Through rough endoplasmic reticulum pathway. Rough endoplasmic reticulum pathway, it is involved in the targeting of secretory proteins. What are the secretory proteins? What are the secretory proteins? Anybody? Name the secretory proteins. Last. Secretory protein. What are the secretory proteins? Examples of secretory protein? That may be hormones, it may be enzymes, toxins. Okay, these are all the secretory proteins. So that means through rough endoplasmic reticulum, most important is that will be your target is your secretory proteins. These proteins have a N-terminal signal peptide sequence. That is very important. In amino acid, two terminal is there. One is C terminal, another is your N terminal. Okay, N terminal and C terminal. So in N terminal, that will be a peptide sequence is there. Signal peptide sequence. This is the signal peptide sequence. Okay. Inside also another near about. After 10 minutes I will ask you what you have learned. So whatever the signal peptide sequence, N-terminal signal peptide sequence near about 70 amino acid composition is near about, length is, what is the length? Length is near about 70 amino acid, near about 70 amino acid is there. Okay. So out of that 30 amino acid near about here, that peptide sequence, and another protein is there, that will be near about 40 amino acid. Understood? Okay, understood or not? So this is ribosome. So inside another 40 amino acid inside it is there. 40 amino acid is there, near about this will be here 30 amino acid. So total length will be here near about 70 amino acid. Understood? Understood or not? So this is your N-terminal signal peptide sequence. Through this sequence, that whatever protein will be there, synthesis, that protein will be enters to the endoplasmic reticulum. Enters to the into the endoplasmic reticulum. This is the very important part. Okay, first I will just discuss it. So this is your ribosome. I told this is your signal peptide. That signal peptide along with ribosome, it will recognized by SRP, that is your signal recognition protein or signal recognition particle. Understand? So SRP, signal recognition protein, it will be binds to the your ribosome along with signal peptide. Okay? After binding this, what will happen? After binding this, it will enters or it will dock to the your endoplasmic reticulum membrane. It will dock to the endoplasmic reticulum membrane. That is called as translocan. That is called as translocan. Okay? Then after this, what will happen? SRP receptor, that is called as signal recognition protein receptor. That receptor present in the endoplasmic reticulum. Now you tell, already your plasma membrane or cell membrane already is over. Now you can tell that SRP receptor, it is which kind of protein? Anybody can tell which kind of protein? It is a which kind of protein? Anybody? See here, this is the inner membrane, outer membrane. So inner membrane, outer membrane, that protein is totally crossed. Yes? Yes? No. No transmembrane. Anybody? What kind of protein? Any other? Suppose cell membrane is there. In cell membrane, the total layer will be crossed. What kind of protein that is? Integral protein. That will be your integral protein, integral membrane protein. So this is a one kind of integral membrane protein which is present in the endoplasmic reticular membrane of the endoplasmic reticulum. So this receptor, SRP receptor, the translocan or the ribosome and signal peptide along with SRP, they will binds to the SRP receptor. They will binds to the SRP receptor to form a SRP complex. SRP complex. So after this binds, what will happen? That will be, now what I told, that during the protein synthesis, how to be terminated? synthesis, how it will be terminated? Suppose protein synthesis is over, how it will be terminated? Terminated by which signal? Termination signal. What is that signal? By stop codon, appearance of stop codon, appearance of stop codon. Now you can remember, this is this area, when that means when docking is there, translocon formation, docking is there, at that time protein synthesis will stop. Understand? At that moment, when translocon will dock to the membrane, endoplasmic reticulum membrane, that time regime of protein synthesis. Protein synthesis is halted. Understood? Protein synthesis now halted. So now that SRP receptor complex, what will happen? That signal peptide, it will enters to the transport channel. This is your transport channel, it will enters to the transport channel. Then after enters to the transport channel, one enzyme, that is signal peptidase enzyme. Enzyme is signal peptidase. That signal peptidase enzyme, it will degrade or it will cleave the signal peptide and protein. This protein, this is the protein, this is your signal peptide. So now it will, here it will degrade. Understand? It will cut there. After that, that protein, this is the protein, this protein will enter to the inside to the endoplasmic membrane. Understood or not? I will draw one by one, I will explain again. Okay? Okay. This is your signal peptide and this is your, what is this? SRP. Signal recognition particle or signal recognition protein. Okay. What will happen? It will bind to the signal peptide. After binding, what will happen? Here that will be this is your transport channel. Now it will dock here. Now it will dock here. That is called as, once it will dock here, that is called as, this is, what is this? This is your translocon. This is your translocon. So SRP, signal recognition protein, along with your signal peptidase, it will binds and docks. When it will docks or enters, dock at the endoplasmic reticulum membrane channel or channel of the transport channel, that will be called as your, what is that? That is called as your translocon. After here binding, here one receptor is there. What is that? What is that receptor? That is SRP receptor. SRP SRP receptor, and that receptor how it will be, sorry here, and that SRP receptor, it is a what kind of protein? This is a what kind of protein? This is a integral membrane protein. Integral membrane protein. So with that receptor, that translocon or ribosome along with signal peptidase and SRP receptor, everything will be bind here. Everything will be bind with the SRP receptor. Understood? Now after binding all, it forms a complex structure. It forms a complex structure that is called as SRP complex. That is called as SRP complex. You can draw or you can write some points. Okay. Satyanan and Vasudevan, that these things you will not get. Okay. That is your SRP complex. After binding all this, this is known as your known as SRP complex. Okay, SRP complex, that is signal recognition protein complex. Now after this, what I told, that complex will be dock, where it will dock? Dock at the endoplasmic reticulum membrane channel. Endoplasmic reticulum membrane channel. Hello. Okay. So after this, what will happen? What enzyme is there inside the membrane? What enzyme is there? That enzyme is the inside membrane. What is that enzyme? This enzyme is there. Signal peptidase enzyme. That will be your signal peptidase enzyme. Okay. That enzyme, peptidase enzyme, what is the role of that enzyme? What is the role of enzyme? That role, importance is there. That what it will do? See, the total complex is there. With that complex, what is there? Ribosome is there. Signal peptide is there. Along with hidden is there. Hidden, what is that hidden protein? Hidden is there. That protein is present, but it is inside or it is hidden. What will happen? That peptidase, signal peptidase enzyme, once it will dock or it will enter through the here membrane, that will be cut or it will be degrade. To whom it will degrade? That enzyme, it will degrade to the, at what point it will cut or it will degrade? I told total 70 amino acids, near about total 70 amino acids. Protein is 40 amino acids and 30 amino acid is your peptide, signal peptide. So what will happen? That signal peptide and that protein is there it will degrade, it will cut. Understand? Understand? It will cut there. After cutting, what will happen? That protein, it will enters inside the endoplasmic reticulum. At this point it will cut. Understood? So this is, I told, what is this? The total is about near about 70 amino acid. Okay, that hidden 40 amino acid protein is there and 30 amino acid, that is your signal peptidase, signal peptide. What will happen? That this enzyme, signal peptidase enzyme, it will degrade here. So now it will be that your ribosome along with signal peptide, it will clip there and it will go for another cycle. It will go for another cycle. And now this protein or this 40 amino acid, this protein will be now it will enter to the inside the endoplasmic reticulum. Understood? Anybody any doubt? Understood? Again I have to repeat it. Or once I have to repeat or not? Huh? See here, first this is your ribosome along with your signal peptide. Okay? Now what will happen? SRP, signal recognition protein or signal recognition particle, it will binds to the that signal peptide along with ribosome. Okay? Now it is clear. After binding this, what will happen? It will enters or it will dock. Enter or you can enter, you can tell dock. It will dock at the, where it will dock? At the transport channel of endoplasmic reticulum. Now it will dock here. Once it will dock, that is called as translocan. That is called as translocan. Okay. After docking here, one receptor is there, that is integral membrane protein receptor. Integral membrane protein receptor that is present in the endoplasmic reticulum. That receptor is called as SRP receptor. That is called as SRP receptor. That is signal recognition protein receptor. That SRP receptor, what it will do? With SRP receptor, everything that will be binds there. So SRP receptor and that SRP particle, signal peptide, everything will bind here. After binding, it forms a complex-like structure. It forms a complex-like structure that is called as SRP complex. Understood? That is called as SRP complex. Called as SRP complex. So after this, what will happen? That I told, that means that signal peptide will dock here. After dock here, what will happen? That signal peptide one enzyme is present, that is signal peptidase enzyme. Signal peptidase enzyme, that is where it is present. It is present in the inside the near the transport channel. Near the transport channel, inside the, that will be inside the endoplasmic reticulum. Inside, not outside, inside the inner membrane of the endoplasmic reticulum. Endoplasmic reticulum, not mitochondria. This is endoplasmic reticulum. Inside the endoplasmic reticulum. What will happen? That will be that signal peptidase enzyme, it will degrade. It will degrade the signal peptide and protein. That connection between the signal peptide and protein, there it will be degrade. Okay. After degrade, that protein, what will do protein? That protein will be enters inside the endoplasmic reticulum membrane. Understood or not? Understood or not? Now it will enter inside the endoplasmic reticulum membrane. Because our target was there, that protein had to enter inside the endoplasmic reticulum. Two pathways is not there. One pathway is the rough endoplasmic reticulum pathway, another pathway is your cytosolic pathway. Now we are discussing about endoplasmic reticulum pathway. So in endoplasmic reticulum pathway, now that protein enters inside the endoplasmic reticulum. Understood or not? Once that protein will be inserted inside the endoplasmic reticulum, from there it will go to Golgi apparatus. It will go to Golgi apparatus. For what? Packaging and for what? What I told, after protein synthesis, next is here, that will be not functional. It will go for functional protein. What is protein have to functional. After synthesis occur, protein synthesis is finished. After that, it is not biological, it is not active. For biological active, what have to go? Anybody? What have to go? Go for modification. It will go for modification. Go for modification. So where it will go? It will go to the, it will enter to the Golgi apparatus for modification. Okay. After modification there, then what it will do? After modification, it will go to their target organ. Through some vesicles. Through some vesicles. Same thing is there. Okay. Signal peptide and here about 7 amino acid long and signal peptide SRP. That particle is there, it is a ribonucleoprotein, all these things already I have discussed. So after that what will happen? That proteins once enter, then it will go for modification. Okay, after modification it will go for, it will enters to the target site by some carrier vesicles. So what are the carrier vesicles? Different carrier vesicles are there. That is your secretory vesicles, transport vesicles and clathrin coated vesicles. Clathrin coated vesicles. Through secretory vesicles, it will involve in the secretion of protein which is expelled from the vesicles at the plasma membrane. So what are the secretory vesicles? What it will do? At the plasma membrane, just near the plasma membrane or at the plasma membrane, that vesicles will be enters there. And through transport vesicles, it will inserted into the membrane. It will inserted into the membrane. At the membrane, your secretory vesicles, transport vesicles will be into the membrane, and clathrin coated vesicles, that will be, what is the role of clathrin coated vesicles? That will be role, it will go for endocytosis, internalization of protein and transport of proteins to lysosome. Okay, so in endoplasmic reticulum, protein now inside the endoplasmic reticulum enters. After that enters, it will go for modification to Golgi apparatus. Understand? After modification in Golgi apparatus, it will enters to the target organ through some carrier vesicles. What are the carrier vesicles? These are the carrier vesicles: secretory vesicles, transport vesicles and clathrin coated vesicles. Secretory vesicle, through secretory vesicles, it will aid the plasma membrane, and transport vesicles inserted into the membrane, and clathrin coated vesicle, it will done by the, it will did what it will do for that enters inside and that transport to the lysosome. So this is your, so endoplasmic reticulum through vesicles, it will enter to the Golgi complex or Golgi apparatus, and there here it will be modification occurs. After modification through vesicles, it will go to target organ. Okay, this is your secretory protein vesicles. By exocytosis, it will outside, and endocytosis, it will enters, and some of them there enters to the lysosome. Now, distribution of proteins in plasma membrane. How it will be distributed? How it will be distributed? Last bench. How it will be distributed? This is your transmembrane domain. What happens? See here. These are your straight or simple transmembrane domain. See here, N terminal is there, C terminal. This will be intracellular cytoplasmic site. Inside the cytoplasm, most of them you will be C terminal. C terminal is there. N terminal, most of them will be outside the cytoplasm or extra cytoplasmic site. These are the here transmembrane domains. These are your simple or single transmembrane domain. These are the multiple transmembrane domain. Multiple transmembrane domain. Now examples will take that here single. So suppose single. So which is more benefit or more benefit? More fast it will enter. More fast. It will be single. If single domain is there, it will be enters more fast. Speed will be more. If multiple is there or several domains are there, that will be, it will take time. So what will happen? Here some examples are there. Straight domain or the simple transmembrane domain. Examples are there here. Growth hormone receptors. Growth hormone receptors. Insulin receptors. Growth hormone receptor, insulin receptor, these are your occurs through single transmembrane domain. Okay, but what happens here? Multiple or several transmembrane domain. Here example we can take, that is glucose transporters and G protein receptors. These are through how to enter? Through multiple transmembrane domain. Through multiple transmembrane domain. Understood or not? So single transmembrane domain and here multiple transmembrane domain. So single transmembrane domain, it will be here. What are these examples? Examples? Single transmembrane domain examples? Already I told. What are these examples? Insulin receptors, growth hormone receptors, okay, LDL receptors. These are here single transmembrane domain. And multiple transmembrane domain or several transmembrane domain, through this what receptors will enter? Works. These are here glucose transporters and G protein receptors. G protein receptors. So after the endoplasmic reticulum pathway, we will go for cytoplasmic pathway. Cytosolic pathway or cytosolic pathway. Cytoplasmic pathway. Large number of proteins are synthesized in the cytoplasm, and after synthesis they are sorted out and targeted to different locations like mitochondrial nucleus. Now how to enter to the mitochondria? How to enter to the mitochondria? Very important. That is here by mitochondrial matrix targeting signal. This signal is there or receptor is there, that is mitochondrial matrix targeting signal. When this signal will be there, then from cytosol it will enters inside the mitochondria. Okay? With this signal only it will enters to the mitochondria. And in nucleus how it will be enters? In nucleus through your NLS, that is nuclear localizing signal. Nuclear localizing signal. When this signal will be given, then only that protein will be enters inside the nucleus. Okay, that will enters inside the nucleus. That is your nuclear localizing signal. Now very important, due to protein targeting or sorting, what diseases may be occur? What are the diseases may occur due to defective protein targeting? Already this disease is already over. So one is your Golgi syndrome, primary hyperoxaluria, familial hypercholesterolemia, cystic fibrosis and eye-cell disease. Eye-cell disease, inclusion cell disease. So one by one will be discussed. In Golgi syndrome, what will happen? What will happen? Very long chain, one more question may come. Very long chain fatty acids, where occurs oxidation of very long chain fatty acids, where occurs? Peroxisome. That is occurs in the peroxisome. So what will happen here? Congenital absence or severe decrease in functional peroxisome. It may be due to genetical defect, it may be peroxisome is absent, or if it is present, that function will be not occur, non-functional peroxisome. So what will happen? Due to that, very long chain fatty acids will be not oxidation occurs, what will happen that due to non-oxidation, that very long chain fatty acids will be more and more accumulated, more and more accumulated. Then once, and due to that accumulation of very long chain fatty acids, what will happen? That severe neurological deficit will occurs. Severe neurological deficit occurs, and it is very fatal in the first year of the life. If it is not oxidation occurs, that patient may die. Okay, this is your Golgi syndrome. Next, primary hyperoxaluria. Primary hyperoxaluria. What happened here? That most important enzyme, that is glycine transaminase. Glycine transaminase. What is the role of glycine transaminase? Glycine transaminase. Transamination. During transamination, that glycine pyruvic acid will convert it to alanine and glycolate. It will convert it to glycolate. What happened here? If this enzyme is deficiency is there or a defect is there, so what will happen? That oxidation, glycolate oxidation to formate will be impairment. So glycolate will be not converted to formate or formic acid. So due to what will happen? That oxalates, that glycolate or oxalates will be more and more will be deposit in tissue. More and more will be deposit in the tissue. Due to that, stones, kidney stones occurs. Okay, so due to that protein targeting defect, that will be, that enzyme deficiency is there. Due to that enzyme deficiency, it is a protein targeting defect. So what will happen? That more and more oxalates will be accumulated. Where it will be accumulated? Where it will be accumulated? Accumulated in the kidney. And these things you have to remember, that glycine transaminase enzyme, it should be present in the peroxisome. It should be present in the peroxisome, but it is present in the mitochondria. That is no role. It should be present in the peroxisome, but actually it is present in the mitochondria, which have no role. Next is your hypercholesterolemia, that is familiar. So what will happen? That LDL receptor will be defective. Defective LDL receptor. So due to defective LDL receptor through which transmembrane protein or which transmembrane regulator? Single domain or multiple domain? Single domain. That is single transmembrane domain. So what will happen here? So due to defect that, what will happen? That LDL will be not catabolized. LDL will be not catabolized. As a result, that LDL level will be increased, which is due to that level of, or you can tell that is hypercholesterolemia. Okay. Normal level, what is the LDL normal level? Less than 130 should be there, but it will increase to, when it will increase to more than 160, it will be go for high risk. Next year is your CFTR, that cystic fibrosis, very important. Cystic fibrosis. What will happen? That defect of CFTR, that is cystic fibrosis transmembrane regulator protein gene. So CFTR gene will be defective. So due to that defective, what will happen? That chloride channel will be disturbed. As a result, what will happen? That water will be moves out from the lungs and pancreas. So more and more water will be moves out from the lungs. As a result, the production of abnormally thick mucus. So mucus production will be more and more thick mucus. Why? Because water is not there. That more water will be expelled. Okay. So due to that thick mucus, so severe pulmonary infection occurs. Understood or not? This is very important. Cystic fibrosis, your short note question. Cystic fibrosis is your short notes, 5 marks question. Okay. What happened here? which gene is responsible for that cystic fibrosis? That is your CFTR gene. CFTR, that is cystic fibrosis transmembrane regulator gene, regulator protein gene. Okay, CFTR gene. That due to this gene deficit or defect, what will happen? That all the maximum water, most of the water will be moves out from the lungs. As a result, the mucus production will be thick. Mucus production will be thick. As a result, what will happen? Severe pulmonary infection occurs. Okay. Next is your eye cell disease. Eye cell disease. That is inclusion cell disease. Inclusion cell disease. So what happens here? That lysosome lacks in enzymes, but they are seen in blood. So due to that protein targeting defect, what will happen? What will happen there? So what will happen there due to defect? Examples we can take that glycoprotein enzyme. Glycoprotein examples we can take glycoprotein enzymes. One example is also mannose 6-phosphate. Mannose 6-phosphate. So what will happen? That lysosomes accumulate many different types of undegraded molecules. Undegraded molecules will be more and more accumulated inside the lysosome as a product or as a occurs, that is inclusion bodies. Inclusion bodies. That examples we can take mannose 6-phosphate. It is the marker to the target the nascent enzyme to lysosomes. In these persons, the carbohydrate units are not added in the enzyme. I told glycoprotein. Glycoprotein means that protein with oligosaccharides unit will be bind. Oligosaccharides unit will be bind to the protein. So what will happen? Due to that enzyme deficiency, what will happen? That oligosaccharide units will not added to the protein. Okay. As a result, so degradation will be not occurs. So undegraded product will be more and more accumulated inside the lysosome. So that part is your protein targeting and sorting. Just little bit near about your protein degradation. Suppose protein is not folded or misfolded protein. What disease occurs? I told that disease is if that unfolded protein or misfolded protein occurs, that may it cause prion diseases. Prion diseases. If that protein will be not proper function. So degradation by two pathway, one is ubiquitin proteasome pathway, another is your lysosomal pathway. Ubiquitin proteasome pathway, that will be energy dependent, energy required, and in lysosomal pathway protein energy will be not required. So what will happen in protein degradation pathway or ubiquitin pathway? Ubiquitin is a highly conserved protein. Highly conserved protein. This structure. This is your ubiquitin. That this protein is misfolded protein. This protein is your misfolded protein or unfolded protein. That will be, if unfolded or misfolded, it will be not proper function. It should go for degradation. It should go for degradation. So for degradation, how it will be occurs? That ubiquitin will bind to that protein. Ubiquitin, it will bind to that protein. That misfolded protein will bind by the ubiquitin. Then once it will bind to the ubiquitin, that it will be enters to the proteasome. It will enters to the proteasome. Proteasome has 2 units is there, regulatory unit and core protein. Core protein is there. So in regulatory unit, it will enters to the along with your protein and protein along with your ubiquitin. After enters here, it will inside here, it will be in core protein, it will degraded. It will be degraded. So now that misfolded protein will be degraded to the different amino acids. Protein to the ubiquitinol binds to that protein and it will enter to the proteasome. After that it will degrade the misfolded protein to amino acids. Okay. In lysosome, what are be degraded? When that protein sialic acid will be removed. Sialic acid will be removed. Other name of sialic acid, anybody can tell? Other name of sialic acid, that is carbohydrate chemistry. Sialic acid. Other name of sialic acid? What is that? N-acetyl neuramic acid. N-acetyl neuramic acid. That is, that sialic acid will be removed from that protein. That sialic acid will be removed from that protein. Once sialic acid will be removed, that will go for degradation. Otherwise, it will not go for degradation. Okay. So, acylated protein, it will be, that is called as, when it will be enters, it will by the receptor acylo-glycoprotein receptor. In presence in the hepatocyte, when that receptor will binds to that acylated protein, then it will go for internalized protein and degraded in the lysosome. Okay, one cycle, one is here through ubiquitin pathway, and that is energy dependent, and energy non-dependent, that is in through lysosome. And through lysosome, how it will be degraded? When in sialic acid will be removed from that protein. Once sialic acid will be removed from that protein, that will be receptor presence in the hepatocyte, that is acylo-glycoprotein receptor. Through that receptor, that protein, acylated protein will be enters and it will degraded in the lysosome. Due to that, numbers of hormones from plasma and will be, the plasma concentration will be maintenance due to that only. Okay, so everything, protein targeting and degradation over. Have you understood or any doubt is there? Any doubt? Any doubt? Anybody can tell? Any doubt or any slide you have to explain? Anybody can explain one slide? Anybody can explain this slide? That means if you, this slide you can explain, so you can write how that protein targeting occurs. Anybody? Anybody? Can you explain or not? Come. Whatever mistake is there, I can modify. Come here. Come. How the protein targeting occurs inside the through endoplasmic reticulum pathway. Come. That's I want to check how much amount or up to what level you have entered. Tell now. Can I explain it? When yes, it forms translocan. When it will translocan forms? When this signal SRP with signal peptide, when it will dock to the endoplasmic reticulum membrane. When it will dock to that transport channel of the endoplasmic reticulum. Then it is called as, that is translocan. Okay, very good. Next. It binds to the SRP receptor. Very good. Then after binds to the SRP receptor, what it will forms? That is called as? That is called as SRP complex. Very good. That is called as SRP complex. Very good. Then now you tell, that SRP receptor, it is a protein. It is a protein. What kind of protein? It is a integral membrane protein. It is a integral membrane protein. Very good. Now it is on SRP complex. Next what is?
Next, protein. This SRP protein enter into the cell.
Which cell?
Endoplasmic reticulum.
Yes. Yes. It will enter to the inside the endoplasmic reticulum. This is your endoplasmic reticulum. Enters into the endoplasmic reticulum. Signal peptide along with your protein. Signal peptide along with your protein. Now see here. This will be protein and this is your signal peptide. All it will enters inside through the, that means it is the membrane is there, it will cross total membrane. Okay. After enters, what will happen?
Binds the degradation by
degradation occurs. Who will degrade? Who will degrade? Signal peptidase. Signal peptidase enzyme. Where it is present? Signal peptidase enzyme. Where it is present? Inside endoplasmic reticulum. Inside endoplasmic reticulum. See here, signal peptidase enzyme is there. After binding here, see here enters, but still now it is not degradation occurs. Still now it is not degradation occurs. When it will cross that membrane, because that enzyme where it is present? Inside membrane. It is present inside membrane. When that peptide, that signal peptide along with your protein, it will enters inside membrane, then only that enzyme will be degraded. Then only it will degrade. Okay. After degrade, that protein will be inside your endoplasmic reticulum. After that, after enters, it will go for modification. Where? To Golgi apparatus. Then from Golgi apparatus, it will go to target organ. It will go to target organ through various vesicles. Okay, understood? Little bit understood or not? Understood or not? Last? Okay, thank you. Thank you, Abhishek. Thank you all.
So the two nerves we are going to learn today is a smaller content of the nerves, which is the extraocular muscle moving nerve, which is known as abducens, fourth cranial and sixth cranial nerve, trochlear and abducens, which are going to talk. We're going to talk this abducens, sixth cranial nerve first, and then we're going to talk about the fourth cranial nerve, which needs like a little bit understanding on this. The extraocular muscle is concerned, or movement of the extraocular muscle is concerned. So we know that particular eyeball having curve of muscles, which is placed in the orbit, and these particular muscles have got like different projections. So we have this lateral rectus, medial rectus, superior rectus, inferior rectus. So they are straight muscles. They are present on different aspects of the eyeball. That's why we call them as rectus muscle. Along with the head, we have some muscle which has a oblique chords. One of them anchored in the upper side, we call them as superior oblique, and the other one which is inferior oblique. So to move these particular four sets of muscles, we have couple of nerves to supply them, and we have this particular nerve which is third cranial nerve, fourth cranial nerve and sixth cranial nerve. They are dividing among themselves to supply to them and make this particular muscle movement of the eyeball. Now, third cranial nerve is already covered, so we are just like starting with this particular fourth and the sixth. We will be starting the sixth cranial nerve first, and then we will talk about the fourth cranial nerve. Now we talk about this particular abducens, which is the sixth cranial nerve, one of the muscle, one of the nerve which was responsible for the movement of the extraocular muscles, and this particular nerve supplies only one muscle. So the sixth cranial nerve moves one intraocular muscle, which is lateral rectus. So the functions of the lateral rectus is always easy. It moves the eyeball laterally. So it's supplied with a nerve which is known as the abducens, and if you do not have this particular lateral rectus moving, that means your eyeball is going to deviate it or pulled by the medial rectus. So it's an easy thing. Many a times we see this particular palsy of this particular nerve, and this particular palsy leads to a kind of eye defect which is pretty common. We see this particular eye defect in the individual, which is leading to the medializations or of this eye, which is like this. So which may have a congenital problem with this person. You can see this one eye deviated medially. This happens because the lateral rectus of this particular individual is not working, it's not pulling laterally. So this medial rectus is superpowered, pulling this particular thing medially, and this particular eyeball moving on directed on a medial aspects. So it is one of the common clinical nerve injury which we see in a clinical life. We will be seeing many of your individuals will be just like a passing by you having this kind of defects. So that's why this particular defect we are talking at the beginning about amongst these particular two cranial nerves, fourth and the sixth. So starting with this particular sixth cranial nerve, as I've said, this is the abducens cranial nerve responsible for this extraocular muscle movement, and only one muscle is innervated by this particular abducens, which is known as lateral rectus, and it's a purely motor. It also have a proprioceptions related to it, but it's grossly purely motor in action whenever his muscle movement is concerned. Now we divide or discuss this particular cranial nerves whenever this particular nerve is concerned, where from these particular nerves are coming. So you see this, you have a nerve which is coming with a nucleus. We have to know where this nucleus is placed. Then this particular nerve shoots the actions which makes this nerve branch, which forms this particular nerve action which runs through some course. Now both of this particular nerve, which is fourth cranial nerve and the sixth cranial nerve, pierces the dura and reach to the space we call them as cavernous sinus, and going through the cavernous sinus eventually gets inside the orbit or eye, going through the supraorbital fissure. The same thing happens with this particular third cranial nerve as well. So this particular sixth cranial nerve, which has a nucleus which is lying in the pons or dorsal pons, part of the pons. Now in this particular regard, this particular nucleus is placed on an area which is known as fourth ventricle of the brain. Now I do not know whether you have gone through the fourth ventricle system of the brain or not. We have a space in the brain which looks like a rhomboid shaped area, which is a little bit projected region in the brainstem or this particular cerebellum region. So that particular region, rhomboid shaped region, we call them as ventricle of the brain. This is known as fourth ventricle. There are several ventricles or expanded space are there which hold the CSF or contain to it blood vessels, neural tissue, etc., and nucleuses. So this particular space, one of them is fourth ventricle, and on the floor of the fourth ventricle we have this particular nucleus of the sixth cranial nerve in place. Now if you have studied the seventh cranial nerve and the nucleus of it, you will see that encircling on this particular seventh cranial nerve nucleus, we have this particular seventh nerve fibers are coming. So wherever you are talking about the nucleus of the seventh nerve, this nucleus of this particular sixth cranial nerve also comes in. But we do not have to just like a detailed discussion about this particular nucleus. It's placed on this particular floor of the ventricle, close to the part we call them as pons. Then you see this particular actions are coming out. Now these actions which is going anteriorly and climbing up little bit runs through this particular interpedunculaire fossa close to this particular circle of Willis, which is a blood circulation supply to this particular brain. Then it runs through. So this is the course. It's coming from the dorsal pons and this related to this particular ventral to the floor of the fourth ventricle, as we have seen the floor of fourth ventricle, and then it runs a course, we call them as cisternal part. It runs through this particular region which is coursing superiorly and anteriorly within this particular interpeduncular fossa, and then it comes with this particular region of the brain, we call them as pontomedullary groove, between the junctions of the pons. Between the junctions of the pons and the medulla, it is coming out. So this is the midbrain region, this is a pons, and this is brainstem, which is like a midbrain pons and medulla, which is coming down. So this is the sixth cranial nerve coming out of here. So in between the pons and the medulla, it is coming out like this, coursing through it, and then it reaches or pierces this particular dura mater and enters to this particular space. We call them as subarachnoid space, which is going in here, reaching to a space we call them as blood vascular system, which is known as cavernous sinus. So along with this particular cavernous sinus, we have this particular walls of the cavernous sinus is there. So if you study this particular cavernous sinus which is present on the sphenoid bone, and this particular cavernous sinus has a roof, floor, it has a medial wall which is related to this particular sella turcica, and this is a lateral wall of the cavernous sinus. So you can see this particular cavernous sinus which is pierced by this number of nerves which is eventually getting to the eye to supply to it, along with this particular large red structure which is the internal carotid artery. Now if you consider this particular cavernous sinus, this particular region has a roof and this is a lateral wall, and this particular nerves which is piercing through the nerves, they have a particular pattern. See that we have this oculomotor nerve which is placed on the superior part or close to the roof, piercing or running through this particular cavernous sinus. Then we have a small nerve which is fourth cranial nerve which is running on here. The abducens is placed somewhere in the central positions under this particular internal carotid artery. Now having a close underlying relations with this region, so if you have an enlargement of this internal carotid artery or branches of this abducens nerve could be have a trouble or injuries to it, which can lead to this palsy of that particular muscles which is supplied by it. Now other branches which is piercing this particular cavernous sinus, which is like the V1 branch of this trigeminal nerve and V2 branch of the trigeminal. So V1, V2 and as well as this trochlear and this oculomotor piercing principally the lateral half or lateral edge starting from the roof to this particular bottom of this lateral wall of the cavernous sinus, whereas this abducens nerve along with this particular internal carotid artery is placed on the central part of this cavernous sinus region. So as I am saying, this particular nerve coursing through this particular cisterna or this particular space which is lying between this particular brain region after emerging from this particular brain of this pons, runs to this particular brainstem region, coursing superiorly and anteriorly, reaching to this particular pontomedullary groove and then reaching to this particular cavernous sinus by piercing the subarachnoid and piercing the dura and entering to the cavernous sinus. Now once you have this particular course on the cavernous sinus is done, it gets into the orbit running through the supraorbital fissure. So it runs through this particular orbit running through the supraorbital fissure, which is in short we call them as SOF. We have the central part of this supraorbital fissure which is having a ring of tendon which is attached to it. We call them as tendinous ring attachments. Piercing through enters through it. Once it enters, it gets into the lateral rectus muscle and supplies this lateral rectus muscle. So it supplies the same side lateral rectus. That's the thing about this particular cranial nerve. Now, the movement of the lateral rectus we already have discussed about. It moves or pulls the orbit or eyeball to the lateral side. Now, if this nerve and this particular muscle is not working, the medial rectus, which is the opposite side, which is working steadily, which is pulling the eye or eyeball to the affected side, to the medial side, and which happens to in this particular clinical problem. In case of this particular compression stretching which happens here, more commonly we see this tumor and this aneurysm of these blood vessels which are related to blood vessels which is coming from the branches of the internal carotid artery, which could be like branches of internal carotid artery like middle meningeal artery or middle cerebral artery or anterior cerebral arteries or cerebellar arteries they are making, or if you have a fracture on this particular bone which is the sphenoid bone, if it is fractured, it can lead to this particular problem. And as well as if you have an infection to the cavernous sinus which is known as cavernous sinus thrombosis, that can lead to this particular compression of the abducens nerve, and that results in inability to abduct the ipsilateral eye, and this individual have this kind of ocular defect where this eye is moved to this particular medial side because of this unopposed action of the medial rectus, because this side lateral rectus is not working. So given with the picture, you may be given an exam question like this particular person is having this kind of defect or ocular eyeball defect or eyeball movement defect, and which particular muscle is not working, or maybe one step ahead, which nerve actually is not working. So you have to figure out which muscle is working and which muscle is not working, and what makes this kind of ocular defect look like because of this some kind of neural problem. So this is one of the thing which is maybe important thing. And And having said this, now we are going to have the next exam, which is coming from the next year onwards, right? So we will have a national based exit exam, which may be coming with this particular MCQ, just like the thing which is like a MD entrance is having. So the next exam may have this kind of clinical picture questions as well in this particular question pattern, because that question is going to be a fully computerized examination, and we might be seeing the pictures and you have to decode the clinical problem based on that. So this is one cranial nerve, sixth cranial nerve. Now coming to this particular other small cranial nerve, which is fourth cranial nerve, which is known as trochlear, which also supplies this intraocular muscle, and which is again is one of the smallest muscle, smallest nerve, and having longest intracranial course. So the trochlear, which is the fourth cranial nerve, which is like a smallest cranial nerve. As I am saying, if you remember, when we have removed the brain, we hardly could see this nerve present on this particular sections of the brain, or neither we could see this particular nerve when we have removed the brain from this particular base of the skull. So this particular nerve is one of the tiniest nerves, very small, very fragile, and very slender. So one of the smallest cranial nerve, and it has a longest intracranial course. So it has a course which runs through the brain itself for a period of time. Now this particular nerve, which is originates from the nucleus, which is placed in the midbrain, a little higher in position compared to the sixth one, which is coming from the pons. So it is coming from this particular nucleus, which is present in the midbrain. Now this particular remarkable feature about this particular nerve is that once this particular nerve coming from the midbrain, it exits from the dorsal aspect. This is one of the things. So whatever the nerves we have seen exiting in this particular brain or brainstem region, they are coming from the brachial side. So they are present on the front side of the brainstem. This is the only nerve which is exiting this particular brain from the posterior side. This is one of the key feature about this particular nerve. So this particular cranial nerve exiting this particular brainstem to the dorsal side or on the posterior side. Second thing about, once it comes to the midbrain, it crisscross or it decussates. So the fibers of this particular nerve, which is this fourth cranial nerve, they cross with each other. So they go to the opposite direction and crisscross and run through this particular space which is lying in the peduncular fossa, and then they just move up and goes to this particular cavernous sinus, and within this particular cavernous sinus they have the particular locations of this particular nerve we already have seen. Inside of this cavernous sinus you see this trochlear nerve is little bit higher in position. So once it gets out of this particular posterior side of the brainstem, it cross over and then reach to the cavernous sinus, piercing the dura, and then it occupies to the higher or close to the roof position over here, and then runs through this cavernous sinus, and coming through the cavernous sinus they get to this particular orbit with the same opening, superior orbital fissure, SOF, and this particular superior orbital fissure, once they get to this particular muscle which is lying over here on the top of this orbit, which is known as superior orbit, and they start innervating by stripping into several small fibers and supplying to it. Now this particular superior oblique muscle, which you have seen in the orbit class as well, and as well as in the eyeball class, this is lying on the top side of the eyeball. Now the striking feature about this particular muscle is that it is present on the medial side on the upper quadrant of this orbit, lying on the superior plane, and this particular muscle has a change of directions of the fibers or change of course by means of the attachments of a pulley or a kind of a lever which is present on this orbit content. So you see this superior oblique muscle going on a course like this, then it's arching back, moving back in a direction. So that's why it has entering to this particular orbit on the superior temple over here by making a loop or orbit content. So whenever this particular muscle is contracting, superior oblique is contracting, this orbit or eyeball moves down. So because of the contraction which is coming from the top side, this eyeball moves down. So depression of this eyeball happens to take place if you have the contractions of this particular muscle, which is the superior oblique. So, this particular superior oblique is having this particular action in the orbit or eye, which is coming on the superior upper quadrant, is this depressing on the eyeball. Now, the prying functions, which is going to the depressions of the eyeball or making this particular eyeball looking go down, if this particular muscle is having any kind of palsies or inappropriate functions, the person most commonly experiences a double vision. Now, this particular double vision, because of this particular three-dimensional perception is not having or not working properly, the person is looking the diplopia or double vision in an upper dome. That means the person is seeing some object and on the top of or around the object he sees the same thing. We call them as vertical diplopia. That means two vision. The diplopia could be of two different types. One is known as the diplopia in a horizontal axis and diplopia in a vertical axis. So, it may be seeing two things one top of another or maybe seeing two things by next to each other. So, you have to inquire the patient. If the patient is saying I am seeing two objects, you have to ask the patient what the object you are seeing, the same object or not, that is the first thing, and second thing, where exactly you are seeing it, on the top or on the side. So, based on that you can just like figure out it is a superior oblique, inferior oblique which is making this particular diplopia thing happening, or if it is a rectus muscle which is creating this kind of problem. So, the axis of the diplopia gives you a clue this nature of this particular muscle involved in here, which is either vertical diplopia above or below diplopia, or it could be a horizontal diplopia which may be affected in this particular patients. Now, having this kind of diplopia, this particular person who have this kind of problem, they try to adjust it. How they are going to adjust it? They just like to tilt the axis of this particular of the object where they are seeing. If they just like to tilt the head, the diplopia goes down or moves up. So, if they see the thing too, they just like tilt the head. So, once they tilt the head, they can just minimize the axis. So, by tilting the head, they will just like to try to see this thing or cut down these particular aberrations of diplopia and try to see the things single, because that diplopia is the thing that makes the thing blurry. So, this particular person who has this kind of diplopia, we see this particular person coming with a tilted head. So, this particular person may be having some kind of neck injuries or neck error which may be leading to this particular tilting of the head. The other reason of tilting of the head could be this vertical diplopia. So, whenever they are coming with the neck problem, you have to examine with the neck problem, with his muscular problem, neural problem with the neck itself or the refractions of this individual. If you correct this refraction which is coming of this particular diplopia, this particular person is getting back the stabilizations of the neck. So, this is in short about these particular two cranial nerves, trochlear and the abducens, and they are a little bit coarse. So, you have to know the muscle supplied by it, and this particular muscle which is supplied these particular nerves, they are purely motor. That means the general somatic efferent, they are supplying to this particular muscle fibers. They are moving this particular orbit or eyeball to this particular directions to its part. So, lateral rectus and this particular superior oblique which is depressing and pulling the orbit down. And if you have a palsy on them, you have to decode the problem, which cranial nerve is not working and what are the silent problem this particular eyeball is experiencing. Alright. So, this is about this particular orbit and the muscles of this particular orbit and that the eyeball itself, which is entirely covered with the nerve supply to it, and optic it is not, if it is not covered, we will just like see optic nerve more commonly seen coming as an exam question in a physiological domain. So, we just like a focus on the extraocular muscles with the nerve supply supplied by the extraocular muscles, and the optic nerve you emphasize on this particular optic nerve and optic nerve lesions based on this particular tract injuries, what kind of like a vision defect the optic nerve has, which is more commonly, more popularly we see that the physiology exam questions are more commonly been given in the exams. So, that is all for the day. Prepare for your physiology exam, which is from 11. Okay, so have ample time. Good luck.
This topic is serum protein electrophoresis. Serum protein electrophoresis is a technique by which we separate serum proteins. Okay. Now, there are two components. One is called serum, another is called plasma. So here we do not take plasma for electrophoresis. Okay. Serum for electrophoresis. And the first thing is, what is electrophoresis? What is electrophoresis? So we must know the definition of electrophoresis. So electrophoresis is the process by which molecules are separated. Molecules are separated on the basis of their charge under an electric field. Under an electric field. But electrophoresis, types of electrophoresis. Paper electrophoresis. Cellulose electrophoresis. Do you know what it is? Cellulose acetate electrophoresis. Agarose gel electrophoresis. Polyacrylamide gel electrophoresis. Cellulose electrophoresis কিছুই না, ম্যাশড পটেটো, আলু সিদ্ধ করতে হবে। আলুকে এমনি ম্যাশ করলে ফাইন একটা মিক্সার তৈরি হবে, কনসিস্টেন্সি তৈরি হবে, সেটাকে একটা ফাইন লেয়ারে কনভার্ট করে তার মধ্যে দিয়ে আমরা ইলেকট্রোফোরেসিস করতে পারি। এবার সেলুলোজ ইলেকট্রোফোরেসিস। সেলুলোজ অ্যাসিটেট ইলেকট্রোফোরেসিস কি? এটা এক ধরনের পেপার তো সেলুলোজ তৈরি হয়। তা সেলুলোজ দিয়ে সেলুলোজের মধ্যে অ্যাসিটেট গ্রুপটা হয়, তবে নেগেটিভ চার্জ তৈরি। এটা অনেক বেশি কম ইম্পিউরিটিস থাকে, তার জন্য ব্যান্ড ফর্মেশনটা ভালো হয়। এর ব্যবহার আছে। সেলুলোজ যখন ব্যবহার করা হয় না, সেলুলোজ অ্যাসিড ব্যবহার করা হয়। সেলুলোজ অ্যাসিড প্রিপেয়ার ইলেকট্রোফোরেসিস ব্যবহার করা হয় ফর আইডেন্টিফিকেশন অফ সিঙ্গল হিমোগ্লোবিন, HBS, single hemoglobin, single hemoglobin commonly ব্যবহার করা হয়। এবার এই সেলুলোজ অ্যাসিড একটা ব্রিটল পেপার, খুব ভেঙে যায়, সেই পেপারটা কি করতে হয়, বাফারে চুবিয়ে তোর একটুখানি শুধু শক্ত দিয়ে ট্যাঙ্কের উপরে রেখে তারপরে কারেন্ট নষ্ট করে তারপর স্যাম্পল অ্যাপ্লিকেশন করলে এই হিমোগ্লোবিনটা বেড়ে যায়। কিন্তু প্রসেস অফ সেপারেশন হিমোগ্লোবিন ইজ ভেরি টিডিয়াস। আমাদের ব্লাড থাকছে, ব্লাড থেকে হিমোগ্লোবিনকে আইসোলেট করে তারপর অ্যাপ্লাই করতে হবে, দেন উই ক্যান রান ইট ফর ইলেকট্রোফোরেসিস। এবার agarose electrophoresis on the other hand, এটা হচ্ছে ডিএনএ আরএনএ মিউটেট হওয়ার কথা। যেমন সারাম বডিং এর সময়, রোজন বডিং এর সময় ডিএনএ আইসোলেট করছি আমরা, তাই না? তাহলে বেসড অন দেয়ার সাইজ এন্ড চার্জ আমরা সেটাকে আইসোলেট করছি, তাই তো? Polyacrylamide electrophoresis is mainly for proteins. কেন না, এটার মধ্যে আমরা the porosity of the gel এটাকে কন্ট্রোল করতে পারি। We can regulate the porosity of the gel. আমরা পোরের সাইজটা বাড়াতে বা কমাতে পারি by increasing or decreasing by managing the concentration of polyelectron. Now, polyelectron can be also two types. A native page, I say HDS. Native page means where proteins natural structure is not hampered. And HDS miniature HDS page is where protein's natural structure is distorted. It is only now their charge on which the migration depends. Otherwise, we have the shape of the protein উপর depend করতে পারে। কিন্তু যদি চার্জে নিয়ে যাই আমরা, তাহলে সেই চার্জে যদি হবে এবং that will be equivalent to their molecular weight. Lower the molecular weight, higher the migration. Higher the molecular weight, lower the migration. Because now, after denaturing, it has no shape. It has only certain amount of charge, and charge is same for all types of polypeptide. Right? So they will move on the basis of charge, but they will get separated on the basis of their size, molecular weight. So we can precisely measure the molecular weight of the protein by HDS stage. Precisely we can do it. Okay? Now, for medical and clinical purposes, we done another important, that is serum electrophoresis, protein electrophoresis, in order to find out the different components of serum proteins. Plasma proteins and serum proteins. What are the difference between these two? Plasma proteins and serum proteins?
They also without clotting factor.
Without clotting factors. What is extra in plasma? Fibrinogen. The fibrinogen is absent in case of serum. When we allow the blood to clot, the fibrinogen is used up. So we get only proteins except fibrinogen in case of serum. Now when we use the serum for electrophoresis, what are different components? Albumin and globulin. Globulins are of three types. Correct? Now, how to perform these gel slide electrophoresis? For what materials we require? Materials we require, you can write glass slide and we go with grease-free glass slide. Grease-free, GREE, grease-free glass slide. 1% agarose solution. Whatman filter paper for sample application. Whatman filter paper for sample application. And WICK, WICK, faucet, porcelain pot for serum. Porcelain pot. Patient serum. Suitable dye, DYE. Hot air oven. And pickles. For viewing, for many days, there was an instrument which used to be used called densitometer. Nowadays it is not used. Densitometer, by using densitometer, we could measure the density of the color, means how much protein is present. from there we can give a rough estimate of the amount of protein present in the slide. But nowadays we just identify the protein and then sufficient, because for estimation of protein we have many other easier methods and more perfect, more precise methods. So that we do not depend on densitometer anymore for guessing the estimate of the actual protein. Now, how to perform the procedure? Procedure লিখতে হবে, আমরা লিখতে হবে ইন্টারপ্রিটেশন হবে। প্রতিটা শুনো ভালো, আমরা কি করবো? what we do, আমরা একটা গ্রিস ফ্রি গ্লাস স্লাইড যেটা আমাদের তোমরা ওই স্পেয়ার বানাও, সেই একই জিনিস, সেই গ্লাস স্লাইড নেওয়া হবে। গ্লাস স্লাইডে গ্রিস ফ্রি করে আমরা তার মধ্যে 1.6 ml 1% লিকুইড আগারোর সলিউশন খাবো। আগারো যদি দেখি ওটা 60 ডিগ্রি সেলসিয়াসের উপরে টেম্পারেচার নিয়ে গেলে আগারো গলে যায়, আবার 40 এর নিচে নামলে ওটা জমে যায়, সলিড। এটা হচ্ছে প্রপার্টি। তবে আমরা যদি গরম করে এই ঠান্ডার উপরে তাহলে ওটা গলে যাবে, গলে গেলে সেটাকে আমরা বাবল ফ্রি অবস্থায় 1.6 ml এর মতন পার স্লাইডে প্রয়োগ করব। ঠিক বাবলগুলো কি হবে? অ্যাভারেশন আসবে মাইগ্রেশনের সময়, এই বাবল ফ্রি করতে হবে, আমাদের গরম ফুটে না যায়। You cannot allow it to boil. We heat it, become liquid, then we pour it by about 1.6 ml on a glass, which is previously made grease-free. Then we allow it to solidify. After the solidification of the gel, it will form like this, a layer on the gel slide. Slowly you can start drawing this. Yesterday only I drew, so I did not wipe it out. Same instrumentation you can follow. So this is a glass slide. But this is a gel layer. These are the weeks, piece of long cut papers, which will connect the buffer tank with the gel. Buffer tank with the gel. Without the supply of buffer, the gel will soon dry up. Okay, so in order to envisage the gel, you must put the wick. Correct? Now, it is important that the layers of the buffer should be in equal height, and thickness of the gel should be uniform. How to do that? We use spirit levels. Where we work, we adjust the spirit level with the table, and then we place the slide, and then we pour. So that there is no undulation, there is no up and down, and will be uniform thickness of the gel layer. Then, if buffers are not having the same height, then what we can do? We can just tilt the gel electrophoresis apparatus, which is nothing but a tank like this, so that buffers they maintain the same level, same height. Then we put the glass slide. We don't put any sample, put the weight, and then we run it for some time. This is called pre-run. The pre-run allows all the ions to migrate in opposite directions, like opposite stars and ions, so that the whole gel becomes moist with the buffer. Then we take small amount of syrup in that porcelain pot, and we do with the help of a faucet, a small piece, very small. Paper is so thin at the piece cut like a weed. This is like syrup with a chubby, soak for a while, ask the chain, be careful, okay? You will place for here, place for one. Yes, you just like that. Take a piece of it. Clean. Every student is put out. Who all the students are not allowed to put the power. Now, this connected to the power pack, which will maintain a particular voltage and current. So if we maintain the voltage at 220 volt, supplied in our normal regular supply, then we must regulate the amount of current to 1.5 ampere per side. Per slide. If there are 10 slides, then 15 ampere total current should be there. Then per slide we will have 1.5 ampere. Follow? Now, when we send current to anything, we follow this rule. Correct? Ohms. What is R? The resistance of the medium to which the current is being passed. Now here, this is another formula called H is equal to I square RT. What is that? The amount of heat generated when current is passed through the medium depends on the square of the amount of current, resistance of the medium, and time through which current is being passed. Now here, in that case, we have to run it for 45 minutes at 220 volt 1.5 ampere per slide current. When we run it like that, we see that almost by 45 minutes the vibration is over. How do we determine that? Previously, before doing that, we can just run with a dye of little less molecular weight, and we can make out measure the time required for the dye to migrate from anode to cathode to anode. Then accordingly we can set the time for protein electrolysis, so that proteins do not go outside this line or migrate very less. If they migrate very less, then there will be no separation. If they migrate very far, then they will go out of this line. Then we will report there is no protein in the serum. So time has to be optimized, say around 45 minutes. Now, if we run for more time, reduce the current, then run for more time. More time means more heat. More heat means what will happen? This gel will become liquid. It will start evaporating, the liquid part, and it will dry up and it will stop migration. Correct? So we cannot reduce the current and increase the time. We have to optimize both current and time. Accordingly, we have to adjust the volt. So all this is done by an instrument called power bank. Okay, where one either voltage or current we have to keep fixed and the other will keep changing, so that we send minimal amount of current required for separation, minimal amount of time. Follow? Now, after the separation is done, we cannot see anything. The glass slide looks like a normal glass slide. You have to put dye in it, you have to stain it. For that, special stains are available. Different compounds require different types of stains. For this, they can use acid black or gold blue, like that they can use. So, blue, when your blue color stain is applied, and it is soaked in the stain. After staining the stain, the overall the gel will take the non-specific stain also. To remove the non-specific staining, we must go for destaining process. The destaining process involves placing this in a trough containing acetic acid and water solution. and several times after distaining, all the unwanted stains are removed, and gel slide still remains. The gel still remains with the bands of the protein color. Next part is fixation. By putting it on a hot air oven and allowing the gel to dry, the gel will dry up and the proteins will get fixed on the glass slide. Done? Now the glass slide is ready for viewing. Okay? Now we can view it. What we will see? We will see different bands. Now, the protein which will move farthest from the point of sample application is
Like
albumin. Why? Smallest in size. Next will come alpha globulin. Next will come beta globulin. And last will come gamma globulin. The gamma globulin may or may not move from the point of application of sample. Sometimes it might go backward. Correct? So, uncharged. ये buffer level पर हमारे buffer pH होचे 8.6 तो ये 10.2 এর মধ্যে থাকে। No. এবার যদি আমরা দেখি out of these four proteins, now alpha can also be subdivided into alpha 1 and alpha 2. Beta can also be subdivided into beta 1 and beta 2. Okay, but that separation, that condition we are not thinking about now. Alpha, albumin, alpha, globulin, beta globulin, followed by gamma globulin. That's all. Now all these proteins are synthesized from liver, except gamma globulin. Correct? So, albumin can be lost through kidney, through urine, for different diseases. So, there will be different interpretation from this class slide. So, now, first thing we will do the normal slide, followed by the different disease slides. So, this is negative side, this is positive side, this is point of sample application. Okay. Have you all drawn this normal slide? Now, one by one, the disease conditions we will consider. The interpretation part of the whole thing. First thing you can say, what does it show? The amount of albumin is low, rest all are fine. Correct? That means albumin is lost, or it is not produced from the liver. What? Which one? There are two possibilities. That it is not being produced from the liver, or it is being lost through urine. This slide shows what? Decrease in albumin concentration. So what could be the possible reason behind it? By seeing the slide, what could we know? How do you support your observation?
Reduce number of apparatus.
Composition formation. What about alpha and beta? Have they reduced? Reduced production parties? Alright. Had it been liver, affected organ is liver, even the alpha and beta globulin also will be produced less. So in that case, if they are normal, if this is less, if this is due to nephrotic syndrome. Fine. Sometimes low albumin nephrotic syndrome. Sometimes there is beta gamma fusion. As if beta and gamma is not separated properly. It is not our mistake. Chronic infection will show this kind of slide. Beta gamma fusion. Albumin will be normal. Fine. What is missing? It's called A gamma globulinemia. What will be a possible disease as it will get? Is gamma globulin is not being produced. Low immunity. What disease? HIV infection can be a cause of agammaglobulinemia. There are other causes also. Familial agammaglobulinemia. By nature, by birth, the individual is unable to produce gamma globulin much. What else? What is it? What do you understand first thing? Then I will tell you. Because diagonally it is very difficult to show. What do you feel by seeing this? The gamma globulin is normal, but alpha, beta, and albumin are less reduced. So yes, number one. So which organ is affected? Liver. Any liver disease of liver in which this occurs? Cirrhosis. Correct? Right? Now, all produced will be less except gamma. Why? Gamma is produced from B cells. It is not produced from liver, it is produced from B cells in circles. Now, sometimes we get an extra band between beta and gamma, and that is very prominent. It's not faint, mostly it is very prominent. Now, here could be a source of confusion. Why? Because by mistake, if you perform the electrophoresis with plasma, then also you will get an extra band between beta and gamma. In that case, it will be fibrinogen for normal individual. But with serum, there is nothing in between beta and gamma unless the person is having multiple myeloma. What is multiple myeloma? Cancer of B cells. Cancer of B cells. In this particular disease, there is only short chains of the immunoglobulins are produced, and that too they are misformed. They have no function, functionless, no bodily function. So they only increase a load of protein, and due to overload of the protein in the overburden, the protein in the plasma, they appear in the urine. This is called overload proteinuria. Okay, overload proteinuria. Sometimes some protein is very high concentration in the plasma, then that is the reason it appears in the urine. There are different types of proteinuria. Right? This is one type of proteinuria which occurs due to overload of the Baines-Jones protein. Baines-Jones protein can also be identified along with this M band. M band is very, very decisive for these persons having multiple myeloma. But beside this, another experiment can be done with 0.1 normal HCl by boiling above 65 degrees Celsius. It will disappear in the urine, and when you cool it down because it's degree, it will again reappear to Baines-Jones protein. This is the case for urine Baines-Jones protein. This is we are doing for serum. So how to কতগুলো হলো these condition? পাঁচটা হলো, তাই তো? ইলেকট্রোফোরেসিসের এটা ওই 5 মিনিট তোমরা খাতায় লিখো, লিখবে, এগুলো পড়বে. This is important for short courses, although this is part of the practical show how, but theoretically the question আসে. ঠিক? এমনি আবার হচ্ছে ইলেকট্রোফোরেসিস বা পেট এগুলো যেরকম কোশ্চেন আসবে, তুমি সিরাম করে ইলেকট্রোফোরেসিস এবং তার ইন্টারপ্রিটেশন যেগুলো কোশ্চেন আসবে. ঠিক আছে? এটা ডিফাইন্ড সিরাম করে ইলেকট্রোফোরেসিস আমরা সব দিলে আসবে না 5 নম্বর 6 নম্বর. তো এইভাবে এইরকম নর্মাল স্লাইড দেখতে কেমন এখন একটা খেয়াল রাখবে, ইলেকট্রোড ডিরেকশন ঠিক রাখবে, আর নর্মাল সাইড কেমন, ডিজ টাইপগুলো কেমন সেগুলো মনে রেখে করতে হবে. কি বললাম, নেফ্রোটিক সিনড্রোম যেখানে অ্যালুমিন পড়ে যাবে, অ্যালুমিন লস হয়ে যাবে, আর একটা হচ্ছে ক্রনিক ইনফেকশন, বিটা গামা ফিউশন, আর একটা হচ্ছে এইচআইভি, এখানে এই গামা গ্লোবিন হচ্ছে, গামা গ্লোবিন পড়ে যাচ্ছে, আর এম ব্যান্ড. এই সিম্পল, বেশি করবে না, সিম্পল এইগুলো মনে রাখবে. আর প্রসিডিওর বা কীভাবে করে স্বাভাবিক জিজ্ঞেস করে, কিন্তু প্রিন্সিপাল জিজ্ঞেস করে. প্রিন্সিপালটা তোমাদের আমি শুরুতেই বলেছি, ডেফিনেশনের মধ্যেও আছে যে all biomolecules will migrate under a given electric field based on their charges. Now, how do you ascertain, how do you be so sure that these proteins are separated into their charges? By increasing the pH of the buffer, that's all. Increase the pH of the buffer. At lower pH it will be positive charge, at higher it will be negatively charged. pH is, normal blood pH is 7.4, we are using 8.6 to 10.2, very high pH. They all will be positively charged. Normally, aluminum is positively charged, all will be positively charged. In this case, all proteins will be negatively charged. So they will migrate equally towards the anode. Any question?
So
सर पीएच बढ़ाने की ज्यादा नहीं होती
হ্যাঁ বাবা?
पीएच बढ़ाने की एक्टिव पता नहीं
না পিএইচ বাড়ালে পিএইচটাকে বাড়ানোর উদ্দেশ্য হচ্ছে সবকটা প্রোটিনকে আমরা আইডেন্টিক্যাল চার্জে নিয়ে আসার জন্য। যেমন মনে করো কোনো প্রোটিনের হয়তো পিআই যাকে বলে যে পিআই মানে হচ্ছে পিএইচ অ্যাকচুয়ালি প্রোটিন উইথ সো নো মুভমেন্ট আন্ডার ইলেকট্রিক ফিল্ড, ঠিক আছে? তাহলে এর নিচে এরা হবে পজিটিভ চার্জ, এর উপরে হবে নেগেটিভ চার্জ, ঠিক আছে? আমি যদি পিআই যেমন অ্যালুমিনিয়ামের ক্ষেত্রে এটা হচ্ছে 4.6, ঠিক আছে? যদি কম পিএইচ করে কি হবে? অ্যালুমিনিয়াম পুরো হয়ে যাবে, অ্যালুমিনিয়াম গাও মিডিয়া চলে আসবে। কোনো প্রোটিন যাতে না থাকে সেটা করা হয়। অ্যালুমিন মানে কিন্তু একটা প্রোটিন না, অ্যালুমিনস বলা হয়। Protein fraction which is soluble in water and dilute salt solution, like normal solution. Globulin কোথায়? Globulin কিন্তু জলে গলে না, এটা সল্ট সলিউশনে গলে, ঠিক আছে? এরকম ডিফারেন্স আছে। তাহলে অ্যালবুমিনস বলা হয়, যেমন এগ অ্যালবুমিন, ওভো অ্যালবুমিন, ল্যাক্ট অ্যালবুমিন, সিরাম অ্যালবুমিন, হিউম্যান সিরাম অ্যালবুমিন, বোভাইন সিরাম অ্যালবুমিন আলাদা। বোভাইন সিরাম অ্যালবুমিন আর হিউম্যান সিরাম আলাদা। বোভাইন সিরাম অ্যালবুমিন একটা হয়, এগ অ্যালবুমিন একটা হয়, এগ অ্যালবুমিনের দায়িত্ব করি, কিন্তু হিউম্যান সিরাম অ্যালবুমিন সিম্পল প্রোটিন। ওই বাবা, আর কোনো কোশ্চেন? No question. Introduce me what is the function of HDS in HDS page? It's actually a detergent. What does it do? It imparts negative charge to the protein. Each polypeptide will cling to 2 molecules of HDS, however long or short it may be. একটা পলিপেপটাইড চেইন দুটো HDS মধ্যে সঙ্গে তাহলে they will migrate on the basis of charge, but they will get separated based on their size, molecular weight. কারণ চার্জ সবাই সমান, দুটো করেই আছে। কি ডিফারেন্স হচ্ছে যেমন সাইজটা, যার সাইজ ছোট সে তাড়াতাড়ি শুনবে, যার সাইজ কম সে আস্তে আস্তে শুনবে। আর কোনো প্রশ্ন? ক্রোমাটোগ্রাফি পাওয়ানোর টেকনিক নিয়ে কোনো প্রশ্ন? কোনো প্রশ্ন নেই? পড়ো এটা, দেখো এখানে অসুবিধা হবে।
So today discuss about CSF, cerebrospinal fluid. So it is a clear. What is CSF? It is a clear, colorless, transparent fluid, which is without turbidity and without powder. Okay, so clear fluid and that is transparent and no turbidity and it is without powder. Where it is synthesized? It is synthesized in the already physiology we have discussed. So where it is synthesized? It is synthesized in the chloride fractions. This is the see carefully here. See here. Where it is synthesized in choroid plexus? Okay. So choroid plexus in the lateral ventricle and also in the fourth ventricle. See, after synthesis, it will enter to the. So from fourth ventricle, how it will enter to the? What is this space? This space is the. Well, what is this space? Space is this is the subarachnoid space. Subarachnoid space. So subarachnoid space means that space between the arachnoid meter and this is the pia meter. Arachnoid meter and pia meter. So between that area, that is the subarachnoid space. So that CSF will enter from fourth ventricle to subarachnoid space through how it enters? Through foramen brusca and foramen mesenteric. Okay, foramen brusca and foramen mesenteric. Then from subarachnoid space, it will enter to the blood. How? Through venous sinus, through superior sagittal venous sinus. Okay. This is important. How it is circulation? How circulation occurs? So circulation from lateral ventricle, it will enter to the first. It will enter to the third ventricle. So lateral ventricle, third ventricle, how it enters? Through foramen of Monro, also called as, that is, interventricular foramen. Interventricular foramen. Then from third ventricle to fourth ventricle through cerebral aqueduct. Through cerebral aqueduct. And from fourth ventricle to subarachnoid space, how it enters? Through foramen of mesentery and foramen of brusca. The three foramina is there. Foramen of, that is, foramen of mesentery and foramen of brusca. Then rate of formation, nearly about 20 ml per hour synthesis occurs. 20 ml per hour. So nearly about 500 ml per day in adult it will be synthesized. 500 ml per day it will be synthesized. So out of that, but actually how much it will be present? 90 to 150 ml in adult it will be present. 90 to 150 ml. Out of that, 90 to 150, 30 to 40 ml will be present in the only ventricle. In ventricle, it will be present how much? 30 to 40 ml. Okay, 30 to 40 ml. So remaining, so out of remaining, how much is there? 100 to 110 to 120 ml. That 110 to 120 ml will be where it will be present. It will be present in the subarachnoid space. Understand? So total how much amount? 90 to 150 ml. Total. Out of that, 30 to 40 ml will be present in the ventricles. Understand? Remaining 110 to 120 ml. Where it will be present? It will be present in the subarachnoid space. And out of that, 110 to 120, 75 to 80 ml will be spinal part, and remaining will be in the radial part. That will be a 25 to 30 ml. Understand? So total how much? 90 to 150 ml. Out of that, 30 to 40 ml will be ventricle. Remaining will be in the subarachnoid space, that is 110 to 120 ml. In subarachnoid space, out of that 110 to 120, 75 to 80 ml will be in the spinal part, and remaining will be in the cranial part. Understand? So important function. What is the functions of CSF? So it acts like a buffer, fluid buffer. Acts as a fluid buffer, and it protects the brain from shock. It acts like a cushion and prevents the movement of the brain against the skull bone, and thereby prevents the brain damage. So it acts like a cushion. Okay. So and most important is the acts like a buffer. Fluid buffer. So from damage, brain damage, it will help prevent the brain damage. CSF is the medium through which many substances, particularly the nutritive substances and waste materials, are the exchanged between the blood and brain tissue. Understand? This is very important function. So nutritive substances, how will it enter? It should be exchanged between the blood and the brain tissue. Who is the medium for that? That medium, I think that is only CSF. Understand? Only CSF is the medium through which only that nutritive substance will be enter from brain and it will out of brain. Okay, so between blood and brain. So who is the medium for extra CSF? This is very important part. Okay. How will we collect the CSF? How will we collect the CSF? Between how many lumbar vertebrae is there? How many lumbar vertebrae? 5 is there. L1 to L5. So always the needle should be put between the L3 and L4. Between the L3 and L4. Okay. Can you tell what is the structure? These are structures. What is this? See here, the tail-like structure. This is spinal cord, okay. In spinal cord, this tail-like structure, what is this?
Nerve.
What is the name of this the tail-like structure? Suppose guru जो बीच में लास्ट है ना, ये गुरु ताकि चुनो तो वही गुरु की जो suppose it is spinal cord, वो last part की the hair-like structure, what is that name? What is that name? Coda equina. Coda equina. These are the tail-like structure. These are the coda equina. Okay, yes, that is the coda equina. So now what is it? See here, very carefully. See between you are taking how they are collecting between the L3 and L4. Why? What is the importance between the L3 and the L4? Why not other part?
Space
one is space is more. Okay, very good. That is acceptable. Another importance?
spinal cord intact have only injury however.
Yes, very good. Spinal cord will not injury. If injury will be a coda equina can be regenerated. Understand? This is the most important. One is thickness is there, another is the suppose that needle will be touched here coda equina. If it is damaged, so that coda equina can be regenerated. That's why taking between the agitated. Understand? Next, this is already you have finished. So what is the composition? Mostly water is 99.13%, solid will be 87%, and what are the organic substance and inorganic substance? Organic substance are the protein, amino acid, sugar, cholesterol, urea, uric acid. These are all organic substance. Inorganic substance: sodium, calcium, potassium, magnesium, chloride. These are all inorganic substances. This is very important. Very important. So first, the color and appearance, it should be clear and colorless and no coagulant, no turbidity. pH, what is the pH? pH of the CSF will be 7.4 to 7.6. 7.4 to 7.6. Specific gravity will be 1.006 to 1.007. And what is the CSF pressure? This is very important. CSF pressure. What is the CSF pressure? How much it is there? It is 100 to 200 mm of water. Okay, CSF pressure should be between 100 to 200 mm of water. And what is the protein content? Protein mostly will be albumin 15 to 45 mg per dl. Milligram per dl. What is serum protein? Serum protein is gram per dl. Understand? Serum protein. When we are taking serum protein, that will be gram per dl. But when that protein will be counted in CSF, that will be milligram per dl. Okay, that is 15 to 45 mg per dl. That is mostly albumin. And globulin, generally globulin will be not there. But then Pendish test will be negative. That will do by Pendish reagent, that will be negative. And glucose content, how much glucose will be there? That will be 45 to 100 milligram per dl. 45 to 100 milligram per dl. Near about, just not exactly, but near about to blood glucose. Near about. And chlorides will be 120 to 130 milli equivalent per liter. urea will be 20 to 40 mg per dl. Calcium 5.5 to 6 milligram per dl. This CSF calcium will be less than or more than serum calcium. Less than or more than? Hello. Serum calcium, what is normal serum calcium? Normal serum calcium level 9 to 11 mg per dl. 9 to 11 mg per dl. But here calcium level will be 5.5 to 6. That means less than the serum calcium. Cholesterol will be very little bit. Inorganic phosphates will be 1.5 to 2.1 mg per dl. Urea 20 to 40 mg per dl. Immunoglobulins mostly present in the IgG and IgA, and lymphocytic will be present 0 to 4 cubic mm. So total volume of CSF will be 100 to 150. So two things you have to keep in mind. So total volume is 100 to 150 ml, and pressure will be 100 to 200 mm of water. Okay. Now, what are the pathological conditions? Which color CSF? What is the color of CSF? Colorless. But when it will be red color, that means it is case of subarachnoid hemorrhage. Okay, subarachnoid hemorrhage and ventricular hemorrhage. Okay, in case of ventricular hemorrhage and subarachnoid hemorrhage, the color of CSF will be red color. And when it will be yellow color in case of spinal tumor. If tumor and tumor occurs in spinal, then it will be the color of CSF will be yellow color. Okay. Then turbidity, when turbidity in case of meningitis, and without coagulum, but when coagulum will be there in case of spinal tumor and tubercular meningitis. In these cases, so the changes of color of CSF will be here. It may be red color, yellow color, and it may be turbid and it may be coagulum in these pathological conditions. Now, glucose content. Content of glucose. The glucose content will decrease in case of meningitis and often disappears completely. That means total glucose will be disappear. In which case? In case of that is meningococci, pneumococci, and streptococci, staphylococci. In these infections, that all glucose will be disappear. And reduced, that is near about 10 to 40 mg per dl in case of tubercular meningitis. And in which case it is increased in case of diabetes mellitus. Because diabetes mellitus blood sugar also increased, that will be enters there and that's why it will be increased. Then fluorides will be decreased in the meningitis. Okay, the reduction is generally more marked in the tuberculosis. In tuberculosis level, it will be less than 102 milliequivalent. Okay, so in case of tuberculosis, meningitis, that will be very dangerous. It will be reduced the level of fluoride. CSF, in which case it will be increased in case of hypertension and renal disease. In case of hypertension and renal disease will be increased. Examination of CSF total protein and specific proteins is used primarily to detect either increased permeability of the blood CSF barrier to plasma proteins or increase the intrathecal synthesis of immunoglobulins. So in which cases that permeability will be increased? The permeability will increase in case of high intracranial pressure. When intracranial pressure will be increased, that time the permeability will be increased. Okay. So in case of brain tumor, that is another one is the intracerebral hemorrhage and trauma due to trauma. Inflammation associated with bacterial or viral meningitis encephalitis and polymyelitis. Premature and full-term neonates have higher permeability. Okay, higher permeability. So high concentration of total CSF protein compared to the healthy adults. So compared to healthy adults, in neonates, those are preterm and term, full term. Then they have the higher permeability. Intrathecal synthesis of immunoglobulins, in particular IgG. Proteins were rated to increase near about 50 to 400 mg per dl in case of tubercular meningitis. Highly increased. Moderately increased 50 to 100 mg in case of benign lymphocytic meningitis, hypertension and cerebral hemorrhage. In these cases, moderately increased, that near about 50 to 100 mg. But highly increased. In which case highly increased? In case of spinal tumor. In case of spinal tumor, so it may goes to 1000 milligram. 1000 milligram per day. In case of brain tumor, total proteins will be increased here about 400 to 800 milligram per day. Calcium is much lower than that of blood serum because much lower protein content. That is 5.5 to 6 mg per dl. The level of calcium reduced. In which case reduced? In case of tetany. In case of tetany, that level of calcium will be reduced. To a decrease in the physiologically active calcium of the plasma. So calcium in which case reduced? In case of tetany. Okay, finished. Okay, any doubt? Regarding CSF, any doubt? सब सारे को बुझते पारले একদম তো গিয়ে আজকে পড়ে নিন। Okay, today we go and today finish it. CSF it may come as a your short note question. Okay. This slide is very important. Okay. This is very important. This slide is important. Product complaints. Okay. आप किसी जिज्ञेस करो जो बुझा रहे थे ठीक है ओके थैंक यू